Yeah, so as just said, I'm just starting my second year. I'm doing a joint PhD actually between the University of Grenoble in France and well, the University of Swansea here. In France, my supervisors are Laure Gallien and Sébastien Lavergne. And here in Swansea, it's Will Allen and Luca Berger. Yeah, I wanted to talk today a bit about the general idea of my PhD, and then the first results of the first chapter, and that will pertain to chameleons, and which is why I entitled my talk "Life History Traits and Long Distance Dispersal Outcomes: The Success of Fast-Paced Chameleons." At the core of my PhD are actually those two questions: why are some species better colonizers than others? And why do some species have large ranges while others do not? And to attempt to answer this question, I'll be looking at the role of dispersal and life history traits in the outcomes of long-distance dispersal events. My PhD as a whole is divided in three parts. The first part, first chapter, I'll be looking at the past and more particularly at the role of traits in biogeographic histories of clades. In the second part of my PhD, I'll be looking at the present, and so I'll be comparing the results of the past to the role of traits in biological invasions that we can see today. And in the last part, I'll be looking at challenges and opportunities in conservation. But so as I said before, I would like to focus today on the first chapter of my PhD, where I'm looking at the role of traits in biogeographic histories of clades. The ultimate goal is to look at many clades at the same time and to kind of find general patterns. But to start with, I chose a single clade, which is the family of chameleons. And so that's what I'm going to talk about now. First of all, I want to give you a quick overview of the biogeography of chameleons. Then I'll be talking about bioregionalization, the paleo reconstruction of biogeoregions, and the fast versus slow life history strategy. And I will conclude with the next steps that I want to undertake in this part of my PhD. In the family of chameleons, there are 204 species that occur mainly in Africa and on Madagascar. But there are also a few species that live on islands in the Indian Ocean, such as the Seychelles and Socotra, which is just off the Horn of Africa, and some chameleons even dispersed as far as to India. And so recent research suggests that this pattern is not the result of vicariance, but it's the result of long-distance dispersal events. And so the question that I want to answer is, why did some chameleon species manage to successfully disperse while others didn't? And so one hypothesis is that traits, more particularly life history traits, play an important role in those patterns. You can see here that there's quite a diversity in chameleons. Some chameleons only live half a year, others 12 years, and they vary a lot in size as well. On the left-hand side, you see the smallest chameleon, which is just as big as your thumbnail, about 18 millimeters long, the Brookesia micra. And on the right-hand side, you can see one of the biggest species of chameleons, Fucifa ustaleti, which has a snout length of approximately 24 centimeters. And there's also quite a variation in clutch sizes. They go from 2 to 52, and clutches per year vary also between 1 and 4. And so one hypothesis concerning long-distance dispersal events is that fast species, so species that produce a lot of offspring and quite frequently, are better at quickly establishing populations and therefore better at successfully dispersing, for example, across the ocean. And so how am I actually going to look at the role of traits in the biogeographic history? I am going to use a model, which is, well, it's not really one model, it's several models, but it's in an R package, which is called BioGeoBears, that was developed quite recently in 2013. And this model actually estimates the movement of species in the past between different regions. And to actually run this model, I need quite a bit of data. For example, the phylogeny of the clade, I need species distribution data to calculate the biogeographic regions so that the model can actually estimate where the ancestors lived of extant species. And then I need life history traits, since that's what I'm looking at. And right now, the model can only deal with a binary trait. So I needed to classify species, chameleon species, into fast or slow species. So the first step, as I said, was to define biogeographic regions. There are many different methods to do this. I used phylogenetic beta diversity and a clustering algorithm, which is called UPGMA. And you can see the result of this analysis here. The important part is actually that islands are discovered or recovered as separate regions so that the model in the end can actually reconstruct movements between islands and the continent, for example. What is important to know, to take into account during the geologic history, the Earth didn't always look the same, and regions moved in relation to each other. And so that's something that can be taken into account in this model. And so the next step of my analysis was actually to reconstruct this movement in the past. And you can see here that, for example, in some periods of time, some regions didn't exist. You saw in the middle of the ocean here, the Seychelles, the splitting of India, and the Comoros Islands, they will emerge, they've emerged quite recently. So I'll just let that finish this. There you can see the Comoros Islands that just popped up in orange. And so the distance between those regions and the existence of the regions actually can be taken into account in the model. And the last part of, I guess the preparation was actually to classify species into fast and slow. For this, I used all sorts of life history data. For example, clutch size, gestation time, sexual maturity. And I used a phylogenetic factor analysis. You can think of it as like a normal factor analysis, or a little bit like a PCA, where basically the information of all those different traits is combined onto a single factor. And so that way I could position chameleon species on a slow-fast continuum, where slow species are characterized by late sexual maturity and long gestation time, and fast species characterized by an early sexual maturity and a short gestation time. And so, as I said before, the model can deal with a binary trait. So what I actually did is I did a median split, and then I assigned all those species that were on the slow side of this spectrum to the group of slow species, and the species that were on the fast side of the spectrum to the group of fast species. Yeah, but so how am I actually going to look at the role of traits then in the biogeographic history? For this, I need to explain the base model a bit more in detail. One of the things that the model estimates is a dispersal parameter, which is called D0. So it is basically the dispersal probability of a species between regions. So the probability, for example, of a species to disperse from Madagascar to India. This dispersal probability can be weighted by distance, because obviously the probability is lower for a species to disperse to India from Madagascar instead of the Comoros Islands, which are quite a bit closer. And so to take traits into account, basically what happens is, depending on whether a species is considered fast or slow, this dispersal parameter will be multiplied by either m fast if it is a fast species, or m slow if it is a slow species. The model cannot estimate both parameters at the same time, both m fast and m slow. So what we actually did is we fixed m fast to 1, which means that the dispersal probability of fast species is equal to the general dispersal probability in the base model. And so the question is actually, what is m slow? If m slow is bigger than 1, that means that slow species are actually better dispersers than fast species. If m slow is between 0 and 1, that means that d0 will be multiplied by a value between 0 and 1, and so the dispersal probability of slow species will be actually lower than that of fast species. And so this is my first result of the PhD. M slow, you can see it on the right-hand side is approximately 0.38, which means that fast chameleons in the past were actually more likely to successfully disperse than slow ones in the past. And so that's kind of a confirmation of the hypothesis that we had at the beginning. But of course, it's not the end of the thing, because next thing that we want to look at, or that I want to look at, is actually things like phylogenetic uncertainty. The whole analysis that I've done so far was done on the consensus tree, the consensus phylogenetic tree, but there's quite a bit of uncertainty concerning the divergence time estimates. And so the next steps are to rerun those analyses, but on a set of different trees, to actually assess the variation of the M-slope parameter, to see if that's actually a robust result. And as I said at the very beginning of this little presentation, the ultimate goal is to rerun this whole analysis on many different clades to see if it's not just a pattern in the family of chameleons that fast species are better dispersers than slow species, but if it's actually a general pattern across all animal species, for example. And so those will be the next steps of my PhD. Thank you for your attention.